ones that I've assigned you, sections 1, 2, and 3, are yesterday, today, and tomorrow. And that's in your WAG. And so basically they just kind of go through each of the types of genetics. And so you can figure it out. So let me know. This is stuff that I kind of assume that most students have from previous classes. And so I don't really spend a whole lot of time on it. Hang on a second. There we go. Okay. So the first one I want to just do is Mendelian. So Mendelian genetics. So you need to get used to that term. Mendelian genetics are normal. Like there is a dominant and there is a recessive. So remember, we're talking about like DNA and chromosomes, right? So we're talking about when you have like one chromosome number 1 from mom and one chromosome number 1 from dad. And on that, they have a gene somewhere in there. Like we did on Monday, we looked at genes on chromosomes. And then one of them might be dominant, one of them might be recessive. The one you got from mom might be dominant, the one you got from dad might be recessive, or they might both be recessive, or they might be both dominant. But that's what we're looking at when we talk about Mendelian. And so these are super typical, like dominant and recessive. These are the easy ones that you probably did in like 7th grade for the first time. Okay. And actually in my pre-AP class, we barely even do these because it's like, that definitely doesn't spell dominant. Sorry. Okay. So let's say we're talking about being able to taste PTC, like the PTC paper that you lay on your tongue and it's like bitter if you can taste it. So if you're a taster, then you're dominant, but maybe you carry the non-tasting gene. And then if you're a non-taster, then of course you'd be that. You put it together. What are my chances of having a child that's a non-taster? 50%. Okay. Mendelian. That's Mendelian. Does anyone have a question about Mendelian genetics? Like, do you know how to get percentages from that? Do you know how to get ratios? Do you remember genotype and phenotype ratio? Maybe I'll do that real quick. Give you a second to write while I write. Okay, so I'm going to put these together in my little boxes. And I try to exaggerate the lowercase t so that I can tell them apart. Okay, so now that I have my little boxes together, I can calculate percentage. It looks like I'm going to have 50% chance that I'll have a child. If I have a kid, 50% chance that our kid would be a taster, so they could taste the weird bitter PTC, and 50% chance that they'd be a non-taster. And I honestly don't even know which one's dominant right now. Like I can't remember. And so basically, so that would be one thing. So one thing you could be asked is, what's the probability? What's the chance that you'll have a dominant or recessive? Okay. And then the other thing they might want is if you could do a ratio. So genotype, these are the actual like type of genes that you have, like what type of gene, what allele do you have available? And so like, that would be like, how many big T do we have? Well, none big T, none in there. We have 2 heterozygous and we have 2 recessive. So the rule is you go from most dominant on the left to most recessive on the right. You can leave off, you want more rules? I love rules. You can leave off the homozygous dominant, but you can't leave off the homozygous recessive. So I always just leave them all on because it's not wrong to leave it on. So I'm like, screw that. I can't remember all the rules. I'm just going to leave, that one on too. But technically you could not have the homozygous dominant on there. I'm not sure why that's a rule. I don't know. Okay. Phenotype ratios, what does it look like? Or in this case, it's not a look because it's a tasting ability. So how is the gene expressed? So on this one, I have 2 tasters, 2 non-tasters. There's not 3 numbers for phenotype ratio because there's only 2 options. You can either taste or you can't. So the phenotype ratio, there's no like 3rd option for Mendelian. Okay, so this is what we're doing right now. Mendelian traits, normal, dominant, and recessive. Okay, questions? This is me not teaching you something new. This is me helping you remember something you learned your freshman year. If that's not the case, you didn't learn it very well or you don't remember it, there's a little ed puzzle that I'll stick in there today that's not going to be due. That's called Mendelian genetics, and you can watch me teach it like I teach it to freshmen. And maybe that will help you if you're like, I'm just not remembering this. Okay, but we actually don't teach the whole thing because we don't have enough time. We have other things we have to do. Okay, So the other options that we're not going to go into today, but I'm going to make a list for you so you know what's coming, because today we're going to spend our time on probability and how to calculate dihybrid crosses and trihybrid crosses and stuff like that, because that's all Mendelian. We're staying in Mendelian today. We're just going to get a little bigger with our Punnett squares. Okay, so the other options that you need to make sure that you're kind of working through as we come to it, because tomorrow is we're going to do a lab with sex-linked traits. If you can have all of this kind of down and ready, we can fly through that lab and you won't have any homework. If not, you're probably going to have to spend the time that we have together in class learning stuff, which means that you don't have as much time for homework, which is fine. Whatever you need is what we're going to do. So sex-linked traits, that's what we're doing. That's what we're focusing on tomorrow. So these are non-Mendelian. These are all non-Mendelian. Let me say that again. These are all non-Mendelian. They're not just recessive dominant. There's something weird about them. Okay. And we'll talk about Mendel in a second and some of his experiments. So sex-linked traits, these are carried on the X chromosome. Okay. So that's one that we're going to do. We're going to do co-dominant and incomplete dominant. Those kind of go together. And then we're going to do multiple alleles. And that, if you remember, was blood type, for example. Okay, and I'm sure you remember these from freshman year at some point. Co-dominant is when both are expressed. So you have a heterozygous, like in the heterozygous situation, you'll see both. So instead of like a red flower and a white flower, when you made them together, like getting red flowers, instead of that, what you would see then is like for co-dominant, you'd have a red flower and a white flower. You'd made them together and you'd get red and white spotted flowers or something like that. Okay. Incomplete dominant is when neither one are dominant. So co-dominant, you see both, like both, whatever is expressed. Incomplete dominant is when none of them are expressed. And instead it's like some sort of blend between the two. Like you have a black cat and a white cat and you cross them together and you get a gray cat. Not black or white is showing. So those are the different traits that we have. Multiple alleles just means there's more than two alleles. Like normally, recessive dominant, there's two alleles there, either big or little. Multiple alleles means you got more options than that. Okay. All right. Those are your list. That's your list of where we're going to be. Hopefully Monday after Thanksgiving, we'll review that and we will be moving on to more exciting things. So that would mean that we'd want to kind of pick it all up by the end of the day tomorrow, if you could. Okay. So there we go. Questions on, I didn't really teach it. Anybody have any comments on that? We'll go over that more tomorrow. Hopefully tomorrow you'll have read a little bit about it or something and the textbook or done whatever you need to do and we can just review. But if not, I'll teach it to you. Okay. And then so today we're focusing once again on Mendelian. Today is Mendel Day in AP Bio. And so Mendelian is based on a fella named Gregor Mendel. You probably know. He did a bunch of work with pea plants, which are like garden peas. And he was a monk. And so he, in Austria, 1800s. And so he just did basically probability is what he figured out. So he figured out independent assortment, which we talked about on Monday, as far as maybe Tuesday, as far as in meiosis, when the chromosomes split apart, when they line up, some line up one way and then the other chromosomes might line up the other way and it's all independent. So he figured independent assortment out, even though he didn't know the mechanism that meiosis was happening. He knew that the genes didn't seem to rely on each other. Like if you had a tall pea plant, then it wasn't like tall went with like green peas every time. It was like the tall was independent from the green, the color of the peas. And so all those traits, he said, they assort independently. Like they don't seem to be like together. And the other thing he talked about, so that's law. You're supposed to know the laws. I should tell you that. Independent assortment. These only work really for they can work for other things, but mostly we do Mendelian with these because some of the other traits are a little bit weird. These only work, I should say that, irrevocably, without any sort of conditions. Independent assortment only works for genes that are carried on separate chromosomes. Like chromosome 1 would have one gene and chromosome 2, whatever's on that, doesn't care what's happening on chromosome 1. Like they're assorted independently of each other. So independent assortment, which means the genes are not dependent on each other. If they're in, they are on, I should say, separate chromosomes. Separate chromies. Okay. And then the other one is, so that's one law of Mendel, one of Mendel's laws, independent assortment. And the other one is the law of segregation, which is a weird one. It's kind of hard to explain. Basically, this one is just like every organism will get like one of each thing. Like it'll be segregated to the organisms. Like if there's a gene for like height and one for like pea color and one for the wrinkly peas or smooth peas or whatever, each of them will be segregated equally to all of those. So it's not like some of them don't have a trait and some of them get like two. It's like equal separation of all the traits. And that's kind of what the law of segregation means. So basically all organisms get a copy of everything. I guess. It's kind of hard to explain. Everything. They go together. Those two laws go together. So basically the genes don't depend on each other to assort, but they all assort to the organisms. Like every organism gets something. And that's what he figured out just working with pea plants in the greenhouse behind the monastery. Okay. Got it? Everybody good? I have to look to my left to see you, so that's why it's kind of weird. Okay, cool. All right, let's work some problems then. Anybody have any questions about that? It's basically what you know about genetics, just had like some names put to it. All right, let's do some dihybrid crosses. Okay, we're going to learn the product rule, multiplication rule. I do it with my freshmen. I don't know if all teachers do it with their freshmen. So I'm going to teach it like you don't know it before. So if you learned it before, bonus. Okay, so basically, let's pretend like we have, do you guys remember this? Let's stop here. Do you guys remember a Punnett square that was like this? Did anyone do that? Where you would be like, okay, we have the tall and round, and then tall and round, and then tall and round, like the different traits, where I guess it's short, whatever. And then you'd put them on this side too. Yes, remember? Okay. Did everybody do a dihybrid cross? Oh, I'm seeing some people shake their heads. No. Okay. And I'm seeing some people shake their heads. Yes. Okay. I'm going to show you. You don't have to make the dihybrid cross on your paper unless you just want to, because I'm going to show you the shortcut way, and we will never make a dihybrid cross in AP biology. Okay. This is the really, really long way to do it. Okay. So if T stands, so we're Mendel now, and we're doing pea plants. We're crossing pea plants. And so T equals tall for the size of the plant. And then so little t, that doesn't even look like a T, equals short. And then the big R means the peas are like round, like you'd think of like in the bag from the freezer section, or, in your can, wherever you find peas. I try not to find peas. They're disgusting. And then wrinkled is like the little wrinkly, like instead of being fat and round, they're like all shriveled, look like a raisin. Okay. It doesn't matter what a pea looks like. Still going to taste bad. So basically what I'm doing here is I'm saying, okay, this parent here was heterozygous for both traits. So that this top parent was heterozygous for both traits. And so I know that what alleles do they have to give? Like if they're going to make sperm or egg or whatever, what could possibly be in those last 4 cells at the end based on what genes they have? And these are the genes that they have, or the alleles that they have, I should say. And so I'm going to basically do some sort of weird little FOIL method where I said, okay, so one of the eggs could have a big T and a big R. That's an option. So there it is. One of the eggs could have a big T and a little r. So there's an option. One of them could have a little t and a big R. There's an option. And one could have a little t and a little r. So there's an option. So I don't know what egg they're going to get, like which one's actually going to be fertilized, but I know that these are the only options we have. And then this one is going to be like big T, big R, little r. Okay. So I've got my big T and my big R here. And then the next option would be big T, little r, because I'm kind of doing a little foil thing here. Then I take my second T. Oh, it's big as well. So big T, big R, and then big T, little r. So this one actually has a repeating thing. I'm going to move that guy out of the way so you can see better. Okay, so this one was, see how far I can go when you can still see it. Who cares? You know what it was. Okay, so then I would just put them all together. Big T, big T. I put my T's together and I put my R's together. Okay, have mercy. You can tell how long this takes. And then at the end, you would just like add it all up. How many you had of each. Phenotype. We don't do genotype ratios of this. Good heavens, no. We just do phenotype ratios. So what are the plants actually going to look like? And guys, I did myself a solid because I had my 1B repeating over there. So I don't have to do this over and over again. Okay, essentially I'm done because it just repeats. Okay, so this one is big, big and big. So that's tall and round. So tall and round is going to be my first option. So I have one of those, 2, 3, 4, 5, 6. So I have 6 of those.
For the paternal side or the maternal side, whatever one you want to call it, for the left-hand side, where did the big T, big R?
Okay, so I pretended like that parent was this. And so then I did it like that instead of being heterozygous. Okay. Does that answer your question?
Yeah.
Okay. Okay, I put 6 here, but technically it should be doubled, right? Because I only did half. So technically this is a 12 because if I were to redo the whole thing again, there would be 12. And then my other option is, tall and wrinkly. Oh, I'm totally writing off of the thing. You should be like, hey, Miss Wilhoit, we cannot see a thing you're writing. So tall and round, I have 12. And then tall and wrinkly, wrinkly, I have 2. Oh, 4, because it should be doubled. So 12, 13, 14, 15, 16. You should have 16 because there's 16 boxes. So I have it all. So basically my ratio is 12 to 4 to 0 to 0 because I don't have any that are short because I had a homozygous dominant here. So I don't have any that are short. Okay. So now I have like an expected ratio. Like if I were to cross these plants, this is what I should get with my little crop of peas. And if I don't get that, then I know that something isn't inherited the way I think it is. I'm leaning you right towards the chi-square is what we're doing there. Because that's my expected, right? I expect to get 12. If I have 16 plants, I should expect sort of to get 12 that are tall and round and 4 that are tall and wrinkly. And I don't, of course, I'm not expecting to get it like on the money. Like every time you breed 2 plants together, you get exactly what you expect. So I have to run a chi-square test to figure out if I'm close enough. We'll do that later, not today. Okay. So, I showed you how to work this thing. I'm going to show you a shortcut so you never have to ever do this again. Is everyone okay with that? Okay, good. I like your thumbs up and your nodding heads. That's why I have your cameras on. Otherwise, I'd be like, everyone okay with that? And then I sit there and stare at myself for a while. Okay, so instead of that, so let's take our parents again. So a plant that is heterozygous for height and round is crossed with a plant that is homozygous for tall height and heterozygous for round. Same one we just did, right? Okay, now instead of doing that big fat Punnett square, I'm going to do 2 little Punnett squares. I can do those really fast. I'm going to do 1 little Punnett square for the height and 1 little Punnett square for the roundness of the pea. Okay, so this one then I would pull just my 2. I'm sorry for my disgusting fingernail. Can't do anything about it. So I would cross those 2 together. So big T, little t, big T, big T. All right. Okay. And then I would cross my rounds together over here, which is big R, little r, big R, little r. And then I'd put them together. And now for the magic. Okay, now you have to know what are my options for my offspring. Okay, so my options could be, let me see if I can move this so I can get it. I'll just move my camera. Oh, please don't fall off. Pick that up, that would be messy. Okay, so now it's like using a microscope. Which way do I turn it? Now I need to know, okay, what are my options? Well, my options are tall and round, tall and wrinkle, short and round, short and wrinkled. Okay, so that would be any options I could possibly have in the great big world of like peas and stuff. Okay, and so now I just have to figure out how to calculate that. I'm going to tell you that I'm going to start multiplying fractions, and I don't ever reduce them. And I know it's weird, and sometimes you're like, I could reduce that. I just don't ever, and it works. And so if I were you, I just wouldn't reduce my fractions. So basically, what are the chances that I would have a tall one? Okay, well, it looks like 50-50 here. So 2 out of 4, see, I didn't reduce it. 2 out of 4 chance that it's tall. So 2 out of 4. And what's the chance that it's round? Round, 1, 2, 3 out of 4. And then I just multiply those together, and I'm officially out of space. So 2 times 3 is 6. Out of 16. Okay, did I do that right? I think so. Okay. You can't see that. Document camera. Okay. So what is my chance of it being tall and wrinkly? Hang on, my brain is clicking because I feel like something's wrong, but I'll figure it out in a second. All right, tall and wrinkly. Well, my chance of being tall is, oh, did anyone catch my mistake? Oh my gosh, I just figured out my mistake.
Yeah, they're all tall.
Yes, I was like, something is wrong. That's my wrong. They're all tall. So 4 times 3 is 12. It's like barely even on the screen. And plus, it's like barely on my whiteboard is the problem. Okay, tall and wrinkly, 4 out of 4 times wrinkly is 1/4. So 4 times 1, that is going to be 4 out of 16. Oh, geez, I needed to plan this better. Short, I got nothing. So since I have 0, when I multiply that, it's 0, right? I mean, 0 times anything is 0. So these are all 0, okay? So 0 out of 16, 0 out of 16. See how fast I just did that dihybrid cross without counting anything? All right, you're going to do one right now. Does everybody have something to write with and write on? Okay, you're going to do a practice one, and you're going to see if you get what I get, and I'm going to erase all this, and unless you need me to keep it on there for a second. If anyone needs me to keep it on there for a second, like wave at me or something. Or put it in the chat. I think there's more of you in here than I can see. I think I can only see 12, and there's 16 of you on here. Okay, cool. Okay, so we are going to do one where you have, oh gosh. Okay, we're going to do one where they're both heterozygous. Okay, so same traits. So okay, here's your clues. Here's your notes to self, because there are ways for you to get these problems right, and there are ways to trip yourself up. And one of the worst ways is not knowing when you get to the end what stands for what. Okay, so I always, even though I've been doing biology for like 100 years, I know I look pretty good, don't I? I know that I'll make a mistake. I'll be buzzing through a test and I will be like, oh yeah, I know what that is. And if I don't make a key for myself, I make a key every time. And so, I mean, maybe you think you're better than me, which you might be, honestly, but I'd make a key. Okay, so let's say we've got, so on the side of your paper somewhere, be like, okay, big T stands for tall, blah, 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 get all that down. Big R is round, little r is wrinkled, and little t is short. Okay, I know what everything stands for. It might take you a second on the test to do that, but in the end, you will pay off by not having to sit there and try to figure out and go back and read the paragraph and all that stuff. These come in paragraphs, and they come with a Mendelian trait with a sex-linked trait, usually. And so you have to do a dihybrid cross with Mendelian and sex-linked, and then figure everything out. And there'll be a paragraph, so if you don't remember what stuff stands for, and then they ask you a question, you have to go reread the paragraph. It's faster to just jot everything down so that you don't have to deal with the mess. Okay, we're going to do two heterozygous parents. So both of them are expressing tall and round, but our hypothesis is that both parents are heterozygous and that traits are inherited in a Mendelian fashion. That's what our hypothesis is. Okay, so that's what we're going to do. Okay, so go ahead and try with the probability method with the multiplication rule. Oh, by the way, you notice I multiplied. That's because they're tall and round, tall and wrinkled, short and there's right? So you always multiply. If in some weird stretch of the imagination they said or, tall or round, then that would be adding. Okay, so I have never, 100% never seen them say or on a test. It's always and. So almost always we just do multiplication the whole time. Just know that if they said or, you would do positive, even positive, good Lord. You do an addition instead of a multiplication. Okay, so this is actually called the multiplication rule for fun. All right, I'm going to do mine here. You do yours there. Don't look at mine until you're done. No cheating. Gosh, can't even with you guys. You will get better at them and you'll be able to shortcut and you'll know what's happening like without having to do every single step. You'll be able to like predict some things. And so it gets faster. But in the beginning, we're going to take it slow. Okay. Once you've got your baby Punnett squares done, then you just have to figure out your probability for them to be. So I just write underneath it. Actually, the easiest way is to just make your list. I wonder if I can fit my list. I'm going to erase my key now and put my list over here. So my list of tall and round, tall and wrinkle. And you also can start abbreviating your list like big T, big R, big T, little r, and stuff like that. But be careful in the beginning until you know what you're doing. Don't shortcut so much that you don't ever learn to do it completely. So there's my 4 options. So I did my Punnett squares. I did my 4 options, and now I'm going to multiply. So what's the chance of it being tall? You'd put that on there. I'm not saying it out loud so that you can think, and then you can look up at mine. If you can see that, my marker is so fat. I need a different marker. I should get a little fine one. Okay. My phenotype ratio for this, from dominant to most recessive with my two hybrids in the middle in any order. I usually do the dominant first, the first dominant trait first, but it doesn't really matter. I've got my answer on my board. If you want to look up and make sure you got your answer too. Any questions? You should have gotten 9 to 3 to 3 to 1. 9 of the dominant, 3 of each of one dominant, one recessive, one dominant, and then 1 of the littles. And that wasn't about a fraction of the time it would have taken you to do that big old Punnett square. Did someone have a question? I heard it unmute.
Is that the genetic ratio?
Yeah, so this is the phenotype ratio.
Oh.
Because it's what they actually look like. So the genotype ratio would be the letters, which would take us, we would never be able to do it. Like we do not do genotype ratios in a dihybrid cross. There would be so many numbers. It would be ridiculous. So once you get past your little single Punnett squares, we'd only do phenotype ratios. We don't do genotype ratios anymore. And guys, when you're ready to do a chi-square test with this, which will be at the beginning of next week, not Thanksgiving, but after, this will be your expected. Okay, this is what I expect if I cross these two together. So that's where we find our expected whenever we're going to do chi-square with this. All right, questions on how to do that? All right, let's do a real quick tri-hybrid. I think you've got it in you. Let's add a trait. Add a trait. Yeah. Look how excited you guys are. Okay, so let's say, okay, ready? Try to write out the genotypes yourself. Okay, I'm going to write them off the screen where you can't see them so I don't forget what I said. Okay, so a plant that is homozygous for tall, heterozygous round, and homozygous recessive for yellow. So make little g's because the dominant is green. So make the letters represent the dominant. Okay, so it's homozygous for tall, heterozygous for round, and homozygous recessive for yellow, which is little g's because green is the dominant. We cross that with a plant that is homozygous short, heterozygous round, and heterozygous green. Okay? Once again, a plant that is homozygous tall, heterozygous round, homozygous yellow, which is the little g's, so recessive, is crossed with homozygous short, heterozygous round, heterozygous green. You got your genotypes. You're going to need 3 Punnett squares. And I was so kind to you, and I made them all different. If one of them is the same, like if you have a heterozygous, you don't have to make 2 Punnett squares. Like you could just be like, it's heterozygous. I'll just use the probability from that 1 Punnett square. But I was so kind to you and made them all different. It doesn't matter which parent's on which side. Like you don't have to have the same one on the left or the right or whatever. It doesn't matter. Okay. So you should have one. Your T's should be a homozygous dominant with a homozygous recessive. Your R's should be both heterozygous. And your G's should be 1 heterozygous, 1 homozygous recessive. Okay, so that's what you should have. And now you have a bunch of options. Am I right? I should put my T's over here. They're in the way. I'm moving my T Punnett square. So now you have tall, round, green, tall, round, yellow, tall, wrinkle, green, tall. Could be here all day. Hello. Then you have to do it all again with short. There they are. Oops. My board is small, limiting what you can see. Okay. I'm trying to. So whenever you multiply fractions, you just multiply across the top. Like you just have to, and then on the bottom, it'll be 4 times 4 times 4, which is 16 times 4, which is, I don't know what that is, 64? Yeah. Okay, I've got a big mess on my board here. I don't have enough space, so hopefully you can kind of see what I'm doing here.
Green is homozygous recessive, right?
Green is the dominant color. That's why we use G's, because that helps you to remember which one's dominant if you can. And so really what happened is when I listed them all out, immediately I had no shorts. And so all I had to do, that sounded kind of funny. So then I had to, I just had to do 4 because none of them were short. And so that was nice. Woo, could have been here all day if I'd had to have all the short too. So then you just multiply across the top. So 4 times 3 is 12, times 2 is 24. So the first one is 24 out of 64. And then 4 times 24 out of 64. And 4 times 2 is 8. 8. So then let's make sure that's right. 24 plus 24 plus 16, 64. Yep. So my ratio that I got is a 24, 24, 8, 8, and then a bunch of zeros. See if you get the same. Can you imagine doing a 64 square Punnett square? They literally take hours if you do it out the long way. Okay. Can I answer anyone's questions?
Yes.
I'm happy to do so.
Why for the short, is it completely 0? Like, why don't we count the round or green?
Because, good question. So remember, we're multiplying, right? So as soon as I have that on there, right there, 0 times 3 times 2 is 0. So I don't have to sit there and do it for all of them. I'm just like, once it multiplies by 0, we're done. There's none of them. Okay. Anybody else? Can I answer a question for anybody? If we're going to end up taking a multiple choice test, which I'm still thinking we're on as far as I know, no, I mean, not maybe for this unit for us, but for the AP test, you're going to want to know how to do this rule, because it is not out of the question for them to give you trihybrid, quadrahybrid. As long as you know how to do it, you can do as many as they give you. But they usually won't ask you to do all of this. They'll just give you one phenotype and say, what's the probability for that one phenotype? Okay, and then you only have to do the math once, which makes it a lot faster. Okay, any questions? Sorry.
For the phenotype ratio, is it just 0? Like, do you put 0 on the ends of the short one?
You can. I'd probably just say 24, 24, 8 to 8. And you can reduce that one if you want to. I just don't ever bother reducing them. But you could just keep on going if you wanted to, long string of zeros. But I usually just put what's there. And honestly, there's not going to be like it's a fill in the blank and you have to say the right numbers in the right order. Like, as long as you know what you're doing, then you usually have to take this information and do something with it. And so then as long as you know what you're doing, you're good. So as long as you make notes to yourself on what's what, it's fine. Okay. Other questions? All right. I'm going to show you how to do your lab. So if you have not gone to the assignment, I hope go to a different tab now. So tab over to if you haven't gone to Pivot Interactives. So if you haven't, I saw maybe 5 from my first hour. I just checked my first hour today and maybe 5 people had. So if you haven't, you need to go to Canvas and there's a Pivot Labs assignment in there. And you need to go there and click on the link so that you can go into Pivot and go click on the link for your hour, okay? And this is Pivot Interactives, and mine looks like this. Yours will just look like one of these. Okay, so in Canvas,